everybody. It's the last one of the unit. This one has to do with nucleic acids. I'm going to kill that bird. Nucleic acids are things that we are used to as being DNA and RNA. RNA, there it is. So what are nucleic acids? Basically, they're made out of carbon, hydrogen, oxygen, and nitrogen, so our chonies. But it also has phosphorus. Phosphorus. So a couple of ways you can think of this. Either one, don't pee in your chonies, or it kind of looks like the word chomp, but with an N, so chomp. So, made out of carbon, hydrogen, oxygen, phosphorus, and nitrogen. The function of nucleic acids is to store and transmit genetic information, so your genes, the way you look, the way you act. And there's also a special class of nucleic acids that their job is to store and release energy. DNA and RNA do not do that. They are this one, DNA and RNA. This thing called ATP, that does that one. Okay, so here's a picture of just a generic slice of DNA showing you all the different things in it. You can see all the rings in it. Looks very similar to glucose, but slightly different. But the main structure is that DNA kind of looks like a staircase or a zipper. All right, let's take a look at specifics. Okay, monomers. So we do have a monomer of a nucleic acid. They're called nucleotides, which is sometimes you guys will have a hard time with the difference between nucleic acid and nucleotide. So nucleotide is the polymer. No, see, now I screwed up. Nucleotide is the monomer. Nucleic acid is the polymer. Okay? It'll take a little bit of practice. So a nucleotide. So there are 4 different kinds, just like there were 20 different kinds of amino acids and like a couple of different types of monomers for carbohydrates. So there's 4 different types of nucleotides that make up nucleic acids. The nucleotide itself is also made up of 3 different things. So this is where it gets a little confusing, as you can see. First thing is it's made up of this thing called a sugar. Now, yes, you think sugar, you think table sugar, sucrose. Well, this sugar is called either ribose. You can always tell a sugar because it ends in OSE. Okay? Sucrose, maltose, fructose, galactose, lactose. Okay? Anything ends in OSE means it's a sugar. And then we have deoxyribose. So it's still ribose, it's still a sugar, but it's without oxygen. So on the sugar, there's no oxygen. This one has it. So we have a sugar, either ribose or deoxyribose. We have a phosphate group. So sticking off to one side, you will see this saying. Basically, it's the element phosphorus with a couple of oxygens hanging off of it, but we usually represent it with just the P. That's a phosphate group. And then we have what's called the nitrogenous base, or nitrogen-containing base. Now the base, that's where the 4 different types come from, because there's 4 different types of bases. I'm going to say these for you so you can practice. Adenine, thymine, cytosine, and guanine. You've probably learned this back in 7th grade, so hopefully it should be at least somewhat of a review. Now, so this is one nucleotide made out of 3 different things. Phosphate stays the same. The sugar, whether it's DNA or RNA, will stay the same. It's the base that changes. Now, DNA has these 4 bases: adenine, thymine, cytosine, guanine. RNA will have a special one, that's a parentheses, a special one called uracil, and it takes the place of thymine. So it'll have a UCG, not a TCG. Okay? So RNA has a special one that takes the place of thymine. Okay, so a nucleotide made up of a sugar, a phosphate, and a base. The base comes in 4 different varieties, unless we're talking about RNA, and then we have 4 varieties, but uracil instead of thymine. Now, if I put all these together in a group, or make a chain of them, then I get the polynucleotide, polymer, the mer being the nucleotide, DNA and RNA. Okay? Now, ATP, that's actually just a nucleotide with a couple of extra phosphate groups sticking off of it, like that. Okay, so let's take a look at the most common ones. Let's look at DNA and RNA. Okay, so here's a nucleotide fancily drawn. This shows all the chemical structure. So here's the sugar right here. Notice it's a 5-ringed sugar. Now this one has oxygen in it. So is this ribose or deoxyribose? It's ribose, okay, because it's got the oxygen in it. Okay, so here's our sugar. here's our base. Our base comes in 4 different varieties. This is one particular variety. Notice how complex it is. We don't need to know the complexity. We just need to know the base goes there. And then we have the phosphate group right here, which is a phosphorus with the 4 oxygens hanging around it, like I told you. Again, we don't need to know necessarily the details of it. We just need to know phosphate. So if I were to draw this simple for you guys to reproduce it, I would do sugar. I would do, something, I don't know, whatever shape you want, base, and then over here, a little circle and phosphate. So there is a simplified nucleotide. Now remember, we said there's 4 different types of bases. So if we look at this one right here, that appears to be, see, NH2, that looks like adenine. So the 4 different bases, they come in 2-ring varieties and 1-ring varieties. The 1-ring varieties has a fancy name called pyrimidines. So the bases that have 1 ring are referred to as pyrimidines. Now the ones that have 2 rings, like these 2 up here, are called purines. So we have pyrimidines and purines. So I can take any one of these 4 and plug it in right here, and that'll give me my 4 different bases, my 4 different nucleotides. Okay, now what if I chain them together? So remember, I've got a sugar, a base, and a phosphate. Now, how they hook together is a little bit different. The bases, you're not going to go like this, phosphate, sugar. It doesn't go like that, okay? The bases are just going to kind of hang out in the middle. So what you're going to do is go phosphate, sugar, phosphate, that's a P, sugar. And for every sugar, there's a base hanging out. Oh, my pen is really annoying me. It was writing nice and smooth, and now it's like, and messing up. Sorry, it's bugging me. Because now it looks like I don't know how to write, and I really do, I swear. Okay, so here's the sugar. Sugar, 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 sugar. Here's the phosphate. Phosphate, phosphate, phosphate. Where is it? There it is. Phosphate. And here's the base, 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 base. So what we get is half of a staircase. Okay, if you kind of imagine it like this, where the half rung of the ladder sticks to the sugar, 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 and then where you put your hands, like right here, phosphate, 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 if you were climbing up the ladder, phosphate, phosphate, and the rungs of the ladder became the base, 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 and base. Okay? Now this is just one single strand. For DNA, it's double-stranded, so I just have another one of these flipped upside down, going the other way. Okay, so DNA is basically just a double-sided strand of the nucleotides. So each one of these is one of the nucleotides. So we can see that I can string them all together like this. Here's where a bond would be. It's just a covalent bond, and then that's how DNA grows. So the polymer is the polynucleotide. Okay. Here's just an example showing you the difference between DNA and RNA. So they're all pretty much the same. They still have a sugar. Sugar, the only difference is that DNA sugar is deoxyribose and RNA sugar is ribose. They both have bases, but whereas DNA has the base T, which is thymine, the RNA has the base U, which is uracil. But other than that, all the rest are the same. So the bases are slightly different. They both have the same phosphate group. So here's the nucleotide for DNA. Here's a nucleotide for RNA. Here's the 4 bases for both. And then when you put them together, this is how they end up forming. So if you notice on this side, it goes phosphate, sugar, phosphate, sugar. So the backbone is phosphates and sugars, and the bases get shoved into the middle. Now the only way to make the bases line up is to flip the other strand over. So if you notice, it says 5' and 3'. The 5's down here and the 3's up here. So you have to actually flip over one of the strands of DNA in order to get them to match up, or else they won't match. And then RNA is just one side. It's just a single strand. So whereas DNA is double-stranded, RNA is single-stranded. So it kind of shows you the 3 fundamental differences between DNA. 1 is the bases that they have. 2 is the strandedness. And then 3 is the sugar that composes them. Okay, so that shows you the difference between DNA and RNA. All right, that's it. that's everything for this section. So go back and make sure you know the 4 different types of macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Under proteins, make sure you know enzymes, and then also how they get put together, how they're taken apart, dehydration synthesis, condensation reactions, hydrolysis, all that good stuff. So go back, re-watch the videos, take notes again, anything you need to help you out. And of course, always ask me for help if you need it. Okay, I will talk to you guys later. Bye.